Hello everyone and welcome to our channel, Mootechie. A special welcome to all the dairy technologists and students with us today. We're going to explore the foundational concepts of fermentation and industrial microbiology. Our presentation begins with the title, Fermentation and Industrial Microbiology. This sets the stage for our discussion on this vital biological process. Let's define fermentation. At its core, fermentation is a chemical process where molecules like glucose are broken down without oxygen, which is called anaerobically. More broadly, the term comes from the foaming observed during wine and beer production, a practice over 10,000 years old. We now know this frothing is caused by carbon dioxide gas, though this wasn't understood until the 17th century. The French chemist Louis Pasteur in the 19th century gave us a more precise definition. He used the term to describe changes caused by yeasts and other microbes growing in the absence of air. He also recognized that products other than alcohol and carbon dioxide could be formed. Next, we consider the anaerobic breakdown of molecules. A key discovery in the 1920s showed that muscle extracts could convert glucose to lactate without air. Importantly, the same intermediate compounds found in grain fermentation were produced in muscle. This led to a major realization. Fermentation is not unique to yeast, but is a common process in glucose utilization by many cells. This brings us to glycolysis. Defined around 1930, glycolysis is the metabolic breakdown of sugar into lactate. It's a form of fermentation where the 6-carbon glucose molecule is split into two 3-carbon pyruvic acid molecules. This process is coupled with the synthesis of ATP adenosine triphosphate, which is the cell's energy currency. Pyruvate can then be processed with oxygen via the tricarboxylic acid cycle or without oxygen, reduced to products like lactic acid or alcohol. This pathway is called the Embden-Meyerhof pathway, named for the German biochemists who elucidated it. So fermentation is now understood as an enzyme-catalyzed, energy-yielding pathway for the anaerobic breakdown of molecules like glucose. These enzymes are in the cell's cytoplasm. The reactions producing ATP and pyruvate are universal, occurring in muscle, yeast, bacteria, and plants. A classic summary is the equation. Glucose yields 2 ethanol and 2 carbon dioxide molecules. Now let's talk about industrial fermentation. These processes start with carefully selected microorganisms and controlled conditions, like specific nutrient levels. The products are incredibly diverse. Yeasts ferment sugars to produce alcohol, glycerol, and text CO_2. Bacteria yield butyl alcohol, acetone, lactic acid, monosodium glutamate, and acetic acid. Molds produce citric acid, gluconic acid, antibiotics, vitamin text B_12, and riboflavin. Ethyl alcohol from starch or sugar is also a key biofuel. Types of fermentation processes. Moving on, we'll look at the different types of fermentation processes. The main categories are microbial cells for biomass, microbial enzymes, microbial metabolites, recombinant products like interferon and vaccines, transformation processes such as those used to make vinegar. Expanding on that, microbial cell mass is used for products like baker's yeast and single-cell protein, SCP. Microbial enzymes are produced on a large scale using fermentation. This is a major advantage. Examples include amylase, protease, lactase, pectinase, and cellulase. Microbial metabolites are divided into two groups, primary metabolites and secondary metabolites, which have different roles and commercial importance. Primary metabolites. Let's examine a table of primary metabolites. This table lists several key products and their commercial significance. Product, commercial significance, ethanol, used in beverages and as a biofuel. Citric acid, has various food industry uses. Glutamic acid, is a flavor enhancer. Lysine, is a feed supplement. Nucleotides, also enhance flavor. Phenylalanine, is a precursor for the sweetener aspartame. Polysaccharides, are used in food and oil recovery. Vitamins, are important feed supplements. Secondary metabolites. Now here is a corresponding table for secondary metabolites. Product, clinical or commercial use. Penicillin, cephalosporin, streptomycin, vital antibiotics. Bleomycin and mitomycin serve as anti-cancer agents. Lovastatin is a cholesterol-lowering drug. Cyclosporin A is an immunosuppressant. Ivermectins are antiparasitic agents. Recombinant products. The field was revolutionized by recombinant products. Recombinant DNA technology allows genes from higher organisms to be inserted into microbes, enabling them to produce foreign or heterologous proteins. Hosts include E. coli, Saccharomyces cerevisiae, and fungi. Animal cells are also used in fermentation systems, though they present challenges like fragility and complex media requirements. Products from genetically engineered organisms are numerous. They include interferon, insulin, human serum albumin, factors VIII and IX, epidermal growth factor, calf chymosin, and bovine somatostatin. Key process design factors include ensuring product secretion, minimizing its degradation, controlling synthesis timing, and maximizing gene expression. Now we'll explore the chronological development of the fermentation industry through a detailed table. This table outlines 6 stages, from before 1900 to the present day. It tracks the evolution of main products, vessel types, process control methods, culture techniques, quality control, pilot plants, and strain selection strategies over time. The table shows the progression from simple alcohol production in wooden vessels to today's sophisticated processes for heterologous proteins and monoclonal antibodies, utilizing computer control and synthetic biology. Major recombinant therapeutic proteins. Another important table lists major recombinant proteins used as therapeutic agents. It groups them by function, such as blood factors, hormones, and cytokines. For each, it shows the protein name, the production system like E. coli or mammalian cells, and its clinical use. For example, factor VIII is produced in mammalian cells to treat anemia. Insulin is made in E. coli and yeast for diabetes treatment. Components of a fermentation process. Now let's break down the parts of a fermentation process. There are 6 basic components. 1. Formulating the growth media for the inoculum and production stages. 2. Sterilizing the medium and all equipment. 3. Producing an active pure culture in large enough quantity for inoculation. 4. Growing the organism in the main production fermenter under optimal conditions. 5. Extracting and purifying the desired product. 6. Disposing of the waste generated by the process. Schematic diagram of a typical fermentation process. Finally, we have a schematic diagram of a typical fermentation process. This flowchart visually connects all the components. It starts with medium formulation and sterilization on the left. It then shows the culture progression from stock culture to shake flask to seed fermenter and finally into the production fermenter. After fermentation, the process splits, biomass can be harvested, and the culture fluid undergoes cell separation. The cell-free supernatant then goes to product extraction and purification, leading to packaging. The diagram also includes effluent treatment, highlighting the importance of waste disposal. This schematic is crucial for understanding how all the individual steps integrate into a complete industrial system. Thank you for joining this comprehensive overview of fermentation and industrial microbiology. We've journeyed from the basic biochemistry of glycolysis to the complex industrial processes that produce everything from food ingredients to life-saving medicines. Understanding these principles is key to innovating in fields like dairy technology. We hope you found this session valuable and informative.